Hello. Very good afternoon to the student who have joined. Can you let me know in the chat box if my voice is audible? And also kindly let me know if you can see the video and the presentation. Okay, thanks for confirming. We will wait till it's 4 p.m. and then we will start today's session. Okay, so it's 4 p.m. Let me start with the introductions. So very good afternoon to all. My name is Mousumi Deb. I'm PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL course of Medicinal Chemistry, and this is our week 5 live session. So today, what we will do is like every live session, we discuss that particular week's course content, and what we do is we solve MCQ questions in the class. Okay, so like every week, today also I will show you around 20 MCQ questions, and each question will have four options. And in the chat box, you have to answer A, B, C, or D. Which of the following will be the correct answer for each question? Okay, and after that, what I will do is I will show you the correct answer and we will discuss that particular question or the particular topic very thoroughly. Okay, so before starting, let me just go through few points. So there will be this kind of problem solving sessions every week from on Friday from 4 p.m. to 6 p.m. Okay, and these sessions are live on Google Meet platform. The link for the particular session is this one, which you have used for joining the class today. This link is same for our every class, so you can just write it down anywhere or you can save it and use the same link to join every Friday class, which is scheduled from this 4 p.m. Okay, so the important point is like if there are any network disruptions happen from my side, please do not leave the session. Okay, I will rejoin as soon as possible, and I am recording the session. That is why I have muted your microphones and also the camera so that I can record the session seamlessly. But the chat box is always open. I am monitoring the chat box every now and then. So if you have any doubt, any query, anything you want to tell me, you can just write it down in the chat box. Okay, because I'm monitoring the chat box every now and then, and since I'm recording the session, the record this particular recording I will be sharing to you after the class. Okay, and also this PPT which I am showing you today, this PPT and the class recording both will be uploaded into the NPTEL website. Where it will be uploaded, I will show you later. Okay, so at the end of the session I will be showing you where it will be recorded, so these things you can find it out there. So the chat box will be used for taking the answers for MCQ question and also for posting your queries. Okay, you can write your doubts, your query regarding any question, any point, you can let me know through the chat box. Okay, so but that's also, if there is any doubt after the class, it is if the doubt persists after the class, I will share my email ID. You can write me down via email. I will try to answer your query via email, or if it is not done, it cannot be done via email, I will just try to answer that in the next class. Okay, so I will share my email ID at the end of the session. So today it is our week 5 live session, and I have already mentioned. So what we do is we discuss that particular week's course content, and how we do it. We will solve MCQ questions. So this type of MCQs will be coming in your assignment, and solving the MCQs during the class will not give you any kind of marks. So you can answer it without any tension that there will be any marking or something. So there is no marks for answering in the question. So if you, even if your answer is wrong, it's completely fine. Okay, but answering is very important. Otherwise, I won't understand that if you are following the class or not. So today it is our week 5 live session, and week 5 is the week 5 assignment which you have to submit next. So what is the topics of week 5? So basically week 5 topics are mainly our nucleic acids at drug target, which are divided into two parts, and also we have some miscellaneous drug targets, and also we have mechanisms in biological chemistry. Okay, so basically the two things we will discuss today. One is the nucleic acids and the use of nucleic acids as drug targets, and also the various mechanisms which are involved in the biological chemistry. So let me start with the today's first question. I hope my voice is audible and you can see the PPT. If there is any problem regarding the audio or the PPT, please let me know in the chat box. So let us check out what is the first question of today's session. So yes, the first question, the problem number 1, it says that camptothecin works as dash. You have to fill in the blanks. Number A, topoisomerase 1 inhibitor. Number B, topoisomerase 2 inhibitor. Number C, DNA guidase inhibitor. And number D, DNA polymerase inhibitor. So camptothecin, it works as what? Which kind of inhibitor camptothecin is? Is it a topoisomerase 1 inhibitor? Is it topoisomerase 2 inhibitor? Is it DNA guidase inhibitor, or is it a DNA polymerase inhibitor? I will give you 2 to 3 minutes time, then I will check the chat box. Okay? A, B, C, or D, which of the following you think is the correct answer? You can write it down in the chat box. I will check your answer, then we will discuss about camptothecin in detail. So basically, I try to solve this, to give this kind of question so that you can easily solve your weekly assignment. And also, when I discuss that particular topic in detail, this will also help you for answering the subjective questions in your final examination. So camptothecin works as which kind of inhibitor or which kind of therapeutic agent? Let me check the chat box. Okay. I have one answer which says that it is number A. Okay. Let me check if number A is the correct answer or not. Okay. So camptothecin works as, yes, number A is the correct one, topoisomerase 1 inhibitor. So camptothecin or CPT, in short, we say it as a CPT. Many portions in the question also, in the books also, sometimes you will find that camptothecin is termed as CPT. Okay, so it is a plant alkaloid. It is the most established inhibitor of DNA topoisomerase 1. So this is the structure of camptothecin. As you can see here, it is a multiple rings are there. It has a kind of planar structure, and also we have a lactone ring system is here. So this is the structure of the active camptothecin. Okay, and when this lactone ring is opened and this carboxylate form portion is formed, so it is the inactive state of the camptothecin. Okay, and this is in the presence of base, we get this kind of your inactive structure of camptothecin. And here we, as you can see, there is a ring which is a quinoline ring. Okay, so the question can, so it's also you can see there is a so this is known as the indeno isoquinol indeno ring. Okay, so topoisomerase 1 is the only target of this drug, and CPT binds neither to TOP1 alone or not DNA alone, but only to topoisomerase 1 and DNA cleavable complex. So then the important molecule we are looking to as is the camptothecin. Camptothecin is a natural product. That's what was extracted from the Chinese bush Camptothecea acuminata in 1966. Camptothecin stabilizes the cleavageable complex that is formed between DNA and the enzyme topoisomerase 1. So by doing this, what happens is that it results a single strand breaks to accumulate the DNA. So now you may remember that topoisomerase 1 and topoisomerase 2 both need the DNA for their function. The difference between topoisomerase 1 and 2 is that topoisomerase 2 cleavage, there is a printing mistake. Topoisomerase 1 cleavages only one strand of DNA. This will be 1. Okay. Topoisomerase 1 cleavage only one strand of DNA, whereas the topoisomerase 2 cleaves both strands, and since the complex between DNA and topoisomerase 1 is stabilized by single break persist, it does not therefore so it does not, and therefore it accumulates in DNA, resulting in inhibition of the proliferation. Okay, so what happens? So camptothecin, so topoisomerase 1 is basically very important enzyme which relaxes the DNA topology during the replication. Okay, the DNA supercoiling, it is relaxed by the a group of family of enzymes which are known as the topoisomerase. So topoisomerase are basically two type, 1 and 2. I already have discussed this topoisomerases earlier in our lecture also. So what happens? The topoisomerase 1, it basically cuts the DNA strand in one strand, only one strand of DNA. But the topoisomerase 2, it cuts the both strands of DNA. So the particular when the topoisomerase 1 cuts the DNA, it makes a bond with the DNA with a particular tyrosine residue. Okay, and this topoisomerase DNA when it is bind to DNA, it is known as the topoisomerase DNA cleavage complex. Okay, so camptothecin. So what happens? Like this cleavage complex later it rotates in somehow and relaxes the DNA supercoiling. But camptothecin is it binds to this cleavage complex and then hinders this particular relaxation of the DNA, thus resulting in inhibition and proliferation. And this is very important that camptothecin is one of the very crucial topoisomerase 1 inhibitors. Okay. So this single strand breaks can be repaired if the drug departs, but if replication is taking place when the enzyme DNA complex is present, an irreversible double strand break takes place, which then leads to a cell death. Semi-synthetic analogs of camptothecin have been developed as a clinically useful anti-cancer agents. Okay, so yes, this camptothecin, it acts as a very important anti-cancer agents, and this is a very specific actually because it only target is the topoisomerase 1. It does not attack or it does not binds with the topoisomerase 2 DNA complex or topoisomerase 2 also. So this selectivity also provides this camptothecin the very importance in the anti-cancer drug discovery. Okay. So yes, number A was the correct answer. Now I will go to the next question. So problem number 2 says, what is the mode of action of proflavin? Your options are number A, alkylating agents, number B, chain cutting agents, number C, antisense agents, and number D, intercalating agents. So what is the mode of action for proflavin? Is it an alkylating agent? Is it a chain cutting agent? Is it an antisense agent or intercalating agent? Let me know in the chat box A, B, C or D which of the following is the correct answer. What is the correct mode of action for proflavin? What is the mode of action of proflavin? Intercalating agent, chain cutting agent, antisense agent, or chain cutting agents? Let me check the chat box now. Okay, I have one answer which says it is number D. Let me check if number D is the correct answer or not. So, the mode of action of proflavin is, yes, number D is the correct answer. It acts as a intercalating agent. So, this is the structure of proflavin. Okay, so proflavin is a topical antiseptic agent used in wound dressing to prevent infections. So this is the intercalating, very important intercalating agent. So as you can see, what is the intercal? So it basically has two portions. If you just look at carefully, in the periphery we have this ammonium ions, okay, which are the polar ions, and also in the middle, what we have aromatic rings here. So these aromatic rings are capable of interacting with the nitrogen interior of the DNA with the van der Waals interactions, okay, and this particular ammonium ions which are in the periphery of the molecule, they interacts with the phosphate sugar backbone with the ionic interaction, as like this. As you can see in the figure, this particular sugar backbone, it is interacting with this ammonium ion with the ionic interaction, and this ring, this planar ring interacting with the this your nitrogen bases with the van der Waals interactions. So, what are the intercalating agents? So, intercalating agents contain flat planar structures that intercalate themselves into the double helix. This distorts the helical structure. At replication, a template that contains an intercalated agent will cause insertion of a random extra base. Then the base pair addition is complete after another round of replication during the intercalating agent is lost. So the examples of the intercalating agents are the indium bromide, proflavin, acridine orange. So intercalating drugs are the compounds that contain planar or heteroaromatic structures that slip between the base pair layers of the DNA double helix. Some of the drugs prefer to approach the helix via the major group, where others prefer access via minor group. Once they are inserted between the nucleic acid base pairs, the aromatic and heteroaromatic rings are held there by the van der Waals interaction with the base pairs above and below. Several intercalating drugs also contain ionized groups which can interact with the charged phosphate groups of the DNA backbone, thus strengthening the interaction. Proflavin is an example of a group of antibacterial compounds that called aminoacridins, which are basically used during World War I and II to treat deep surface wounds. These drugs prove highly effective in preventing the infection and reduce the number of fatalities resulting from the wound infection. Proflavin is completely ionized at the pH 7 and interacts directly with the bacterial DNA. The flat tricyclic ring intercalates between the DNA base pair and interacts with them via van der Waals forces, while the ammonium cations from ionic bonds with the negatively charged phosphate group of the sugar phosphate backbone. Once inserted, proflavin deforms the DNA double helix, prevents the normal function of replication and transcription. Okay. So this is proflavin is one example of intercalating agents. There are ethidium bromide, acridine orange are also there, which basically the mechanism of action is same as the proflavin. So this is the all about the intercalating agents which you need to know and which you need to remember. Okay. Now I will go to the next problem. Problem number 3 says, which of the following is not a microtubule inhibitor? Your options are number A, cisplatin, number B, paclitaxel, number C, vinblastine, and number D, colchicine. Which of the following is not a microtubule inhibitor? Cisplatin, paclitaxel, vinblastine, or colchicine? Again, I will give you some time. Read and then let me know in the chat box A, B, C, or D which you think is the correct answer. Which of the following is not a microtubule inhibitor? Problem number three is it says which of the following is not a microtubule inhibitor? Your options are number A cisplatin, number B paclitaxel, number C vinblastine, number D colchicine. Let me know in the chat box A, B, C or D which of the following you think is the correct answer of problem number three. Okay, let me check the chat box now. Okay, I have got two answers. One is saying it is the number C, one is saying it is the number A. Let me check which of the following is the correct answer, number C or number A. Okay, so the one which is not a microtubule inhibitor is it is number A, cisplatin. So cisplatin is not a microtubule inhibitor. So what are the different drugs that target tubulin? So tubulin is a very important cell component which are targeted in the anti-cancer drug discovery and also in the various your drug as a various therapeutic agents. So basically, what are the drugs that targets tubulin? So we have the vinca alkaloids, which are the vincristine and vinblastine. So these are the natural products that binds with the tubulin, inhibit the microtubule formation, which leads to the metaphase arrest, and it acts as an anti-cancer agents. Okay. Then we have the taxanes. So taxanes, what they do is, so they freezes the microtubules in the metaphase, and tubule molecules fails to depolymerize, and which leads to the cell death. Okay. Then we have the our mebendazole and also the albendazole. So what they do, they blocks the polymerization of tubulin in the intestinal cells, blocks the glucose and glucose uptake, and the gradual immobilization and death of the helminthes. Then we have this grisfosflavin, which binds with the fungal tubulin, impaired the microtubule formation, inhibit the mitosis, which is the fungistatic agent. Okay. Then we have the colchicine. Colchicine binds with the tubulin, inhibit the microtubule polymerization, then impaired activation and migration. Then neutrophils. This reduces the inflammation in gut. Okay, so which it is a very important drug for which targets the arthritis. Then so here as you can see, there are the different microtubule inhibitors. So we have the paclitaxel or paclitaxel, which are the taxane binding. Okay, this is all are these the microtubule inhibitors, but the binding domains are different. Like paclitaxel is a taxane binding, which which uses in the therapeutic as a therapeutic agents in breast, ovarian, prostate, and lung cancer. Then the mode of action is the anti-microtubule depolymerization leading to mitotic arrest. Okay. Then we have this docetaxel, which is also the taxane binding. That cancer treatment used for the breast, non-small cell lung cancer, androgen independent metastasis prostate cancer. The mode of actions is the anti-microtubule depolymerization and the attenuation of the BCL2 and BCL-XL gene expression. Then we have colchicine, which is colchicine binding, which is used in the hepatocellular and prostate cancers. Anti-microtubule polymerization, cell cycle arrest in metaphase. Also, we have vinblastine, which is the vinca binding. This testicular, Hodgkin's and non-Hodgkin's lymphoma, breast and germ cell cancers. And the mode of action is induces the wedge at a tubule interface, causing tubulin self-association into the spinal aggregates, anti-microtubule polymerization and cell cycle arrest in metaphase. Okay, so all these are the microtubule inhibitors, and there are with their binding domains, mode of action, and the therapeutic areas. And this is also very important chart which you have to remember regarding this microtubule inhibition. Okay, now I will go to the next problem. Okay, so one more thing that this particular cisplatin, which I have mentioned that it is not a microtubule inhibitor. So, what is the mode of action of cisplatin? So, the main mechanism of cytotoxic action involves the binding of the cisplatin to genomic DNA in the cell nucleus to form intrastrand and interstrand crosslinks that interferes with the normal transcription and or DNA replication mechanisms and triggers the cytotoxic processes that lead to the cell death. Okay, we will discuss regarding the particular cisplatin in later in our today's class. Okay, now I will go to the next problem. So problem number 4 says doxorubicin is an intercalating agent which belongs to a group of naturally occurring antibiotics. What role does the ionic group serve to enhance the intercalation effects of this molecule? Your options are: number A, it increases the water solubility, allowing the high dose levels to be used. Number B, it interacts with the charged phosphate groups in the DNA to stabilize the intercalation. Number C, it acts as a strong hydrogen bond donor with the nucleic acid bases to stabilize the intercalation. And number D, it only becomes protonated when it enters the cells and so becomes trapped within the cell, leading to the high concentration of the drug. Again, I will repeat the question. Doxorubicin is an intercalating agent which belongs to a group of naturally occurring antibiotics. What role does the ionic group serve to enhance the intercalation effect of this molecule? The options are: number A, it increases the water solubility, allowing the high dose levels to be used. Number B, it interacts with the charged phosphate groups in the DNA to stabilize the intercalation. Number C, it acts as a strong hydrogen bond donor with the nucleic acid bases to stabilize the intercalation. Number D, it only becomes protonated when it enters the cells and so becomes trapped within the cell, leading to the high concentrations of the drug. A, B, C, or D, which of the following is the correct answer? You have to let me know. So, what is the correct answer of problem number 4? Okay, I have few answers. All are saying that it is the number B, I think. Yeah. Let me check if number B is the correct answer or something else. So, the correct answer for problem number 4, yes, it's number B. The role of the ionic groups, which is serves to enhance the intercalation effects of this molecule, is it interacts with the charged phosphate groups in DNA to stabilize the intercalation. This is the structure of the doxorubicin. As you can see, it is have four rings which are joined with each other. Okay, and here we have a side linkage which have this ionic group, so which is basically the ammonium ions. So, doxorubicin is one of the most effective anti-cancer drugs ever discovered, and it belongs to a group of naturally occurring antibiotics called anthracyclines. The first isolated from the Streptomyces fuset in 1967 and contains a tetracyclic ring system, and three of these rings are the planar. The drug approaches the DNA via the major group of the double helix and intercalates using the planar tricyclic system. Okay, so this is the doxorubicin in the DNA structure. So the charged amino group attached to the sugar is also important. It forms the ionic bond with the negatively charged phosphate groups in the DNA backbone. This is supported by the fact that the structures lacking the amino sugar have poor activity. Intercalation prevents the normal action of the enzyme called topoisomerase II, an enzyme that is crucial for replication and mitosis. The mechanism of topoisomerase II involves the formation of the DNA enzyme complex where the enzyme is covalently linked to the DNA. When doxorubicin is intercalated into the DNA, it stabilizes the DNA enzyme complex and stalls the process. Agents such as doxorubicin are referred as topoisomerase II poisons rather than inhibitors, as they do not prevent the enzyme functioning directly. Okay, so there are two terms which are very important. One is the poison and one is the inhibitor. So inhibitors are basically referred to those drug agent which directly inhibits the enzyme for working. Okay, but poisons are different from the particular inhibitors, because the poisons, they do not prevent the enzyme work. Rather, this particular poison, the doxorubicin, which is a poison of this topoisomerase II enzyme, what does it do? It stabilizes the DNA enzyme cleavage complex, and during the relaxation process, because of this stabilization of this DNA cleavage complex, what happens? The this particular relaxation process is hindered. Okay, so this topoisomerase II poison is particularly your doxorubicin. So its target is topoisomerase II, whereas the camptothecin's target is the topoisomerase I. Okay. So just remember this thing, which is very important, that how doxorubicin behaves and the ammonium group, which so this is also similar to kind of the proflavin which we have earlier discussed. There also it has the ammonium ion which are in the periphery of the molecule, which interacts with the sugar backbone of this particular DNA. Okay, so this is almost similar mechanism of action which happens here also. Now I will go to the next problem. Problem number 5 says, which DNA base interacts with the anti-cancer intercalating agent dactinomycin? Your options are number A adenine, number B thymine, number C cytosine, number D guanine. Which DNA base interacts with the anti-cancer intercalating agent dactinomycin? Adenine, thymine, cytosine or guanine? Let me know in the chat box A, B, C or D which of the following you think is the correct answer. Which of the following DNA base interacts with the anti-cancer intercalating agent dactinomycin? Which DNA base interacts with the anticancer intercalating agent dactinomycin? Adenine, thymine, cytosine, guanine. Okay, let me check the chat box now. Okay, so I have answers number D, number D, number D. So three answers I have got. Was all the three are saying it is the number D. Okay, let me check if number D is the correct answer or not. So the DNA base which interacts with the intercalating anticancer agent dactinomycin is, yes, it is number D, guanine. So this is the structure of the dactinomycin. Okay. So another example of an intercalating agent is dextromycin. It is previously called as the actinomycin D. It is naturally occurring antibiotic that was first isolated from the Streptomyces parvulus in 1953 and has been shown an effect as an effective anti-cancer agent in childhood cancers. If you look at the structure of the dactinomycin, so it has this planar phenoxazone system, which is this one, this planar phenoxazone system, and it has also a peptide over here, and which is a cyclic peptide. So the structure is very unique. It has a planar phenoxazone system, also the cyclic peptide in the here also. Okay, so the drug approaches the DNA via the major group of the double helix, and after it intercalates, the planar tricyclic system gets between the two base pairs. As we have seen previously, the charged amino group attached to the sugar is also important, and it forms an ionic bond with the negatively charged phosphate groups in the DNA backbone. It appears to favor the interactions with the guanine cytosine base pairs, that is the GC base pairs, and particular between two adjacent guanine bases on the alternative strand of the helix. So you have a GC interaction, and if you have two Gs, it prefer to bind this type of DNA base pairs. The molecule is further held in position by the hydrogen bond interaction between the nucleic acid bases and the cyclic pentapeptides, as we have looked at earlier. Okay. So this is the DNA intercalation and dactinomycin, and it interacts with the GC base pairs and GG. If there is two Gs also, this all it will prefer to interact with those particular portions. Okay. So now I will go to the next problem. Okay, so we have a little bit also. So the two amino group of this guanine plays a particular important role in interaction. The resulting bound complex is very stable, and it prevents the unwinding of the double helix. So in turn prevents the DNA-dependent RNA polymerase from catalyzing the synthesis of the mRNA, and it prevents the transcription. So the effect of dactinomycin is actively dividing cell is that once it gets into it in the inside and does the intercalate, after the intercalation it forms an extremely stable bond with DNA, and after this complex is formed, because of the complex is so stable, it prevents the unwinding of the double helix, and unwinding, if the unwinding of double helix does not happen, as we know from the previous lectures that the single strand DNA is not produced, which is going to transfer the information to mRNA, and therefore transcription is inhibited, and as a consequence of if the transcription is inhibited, then the cell will stop replicating and stop dividing. Okay, so this is the complete mechanism of action of dactinomycin. So remembering the structure is not much important, but remembering the mechanism of actions of this kind of therapeutic agents are very, very important. Okay. I'm trying to give you the example of each section of the different classes of therapeutic agent, like intercalating agents, topoisomerase 1 inhibitor, 2 inhibitor, and so on. So this you have to remember the mechanism of actions of all the particular agents and with the examples. Now I will go to the next problem. So problem number 6 is, which toxic side product is generated from the alkylating agent cyclophosphoramide? So which kind of toxic side product is generated from the alkylating agent cyclophosphamide? Your options are number A acrolein, number B crotonaldehyde, number C cytosine, number D guanine. Which toxic side product is generated from the alkylating agent cyclophosphamide? Let me know in the chat box A, B, C or D which of the following is the correct answer. Which toxic side product is generated from the cyclophosphamide, which is an alkylating agent? Which toxic side product is generated by cyclophosphamide? Let me check the chat box now. Okay, number A is the given answer. Let me check if number A is the correct answer or not. So the correct answer is yes, it is number A, acrolein. Acrolein is the toxic side product which is generated by the alkylating agent cyclophosphamide. So phosphamide mustard is the main cytotoxic and alkylating agent. So cyclophosphamide basically it is a prodrug. Okay, so it is activated. So this has this kind of structure where R is the CH2CH2Cl, and this is activated by the cytochrome P450 enzymes. And what does this do? This hydroxylates in this position. Okay, this enzyme adds a hydroxyl group in this position, and this hydroxyl group, then what happens? The chain opening reaction is occurred here, and this chain occurring reaction gives rise to this compound, which is the cytotoxic alkylating agent. Okay, or the alkylating agent, which is called as the phosphoramide mustard, and also we have a side product of acrolein. So cyclophosphamide is the most commonly used alkylating agent in the cancer chemotherapy. It is a non-toxic product which is converted in the body into the active drug. Okay, so this is also very important to remember. The cyclophosphamide is a prodrug which is activated by the cytochrome P450 enzymes in the body. The metabolism takes place in the liver where the cytochrome P450 enzymes oxidize the ring. The ring opening then takes place and the non-enzymatic hydrolysis splits the acrolein from the molecule to generate the cytotoxic alkylating agents. The nucleophilicity of the nitrogen is tempered by it being it a part of the phosphoramide group, and so the active agent is more selective to the stronger nucleophiles such as guanine. Cyclophosphamide is itself relatively non-toxic and can be taken orally without causing damage into the gut wall. It was also hoped that the high level of phosphoramide phosphoramidase enzyme also present in some tumor cell will lead to the greater concentration of the alkylating agent in these cells and result in the selectivity of action. Unfortunately, the acrolein released is sometimes proven to be toxic to the kidneys and the bladder. One possible explanation is that acrolein alkylates the cytosine residues in the cell proteins. Certainly, toxicity can be reduced by co-administrating the sulfhydryl donors such as the N-acetyl cytosine or the sodium 2-mercaptophan sulfonate or mesno, which interact with the acrolein. Okay, so this particular acrolein it seems to be toxic to the several our organs like bladder, kidneys, and so on. So one way to reduce this toxicity is to co-administrating the cyclophosphamide with this sulfhydryl donors molecules, which will bind with the acrolein and thus reducing the toxicity. Okay, so this is the mechanism of action of cyclophosphamide, and which is a very important alkylating agent. Now I will go to the next problem. So problem number 7 says the major intercalating species in bleomycin is dash. Your options are number A thiazole ring, number B imidazole ring, number C bisthiazole ring, number D indole ring. What is the major intercalating species in bleomycin? Thiazole ring, imidazole ring, bisthiazole ring or indole ring? Let me know in the chat box A, B, C or D which of the following is the correct answer. What is the major intercalating species in bleomycin? Thiazole ring, imidazole ring, disthiazole ring, or indole ring? So what is the major intercalating species in bleomycin? Thiazole ring, imidazole ring, disthiazole ring, or indole ring? Let me check the chat box. Okay, so I have few answers, number A, number C, number A. So I think I have two number A and one number C. Okay, let me check which of the following is the correct answer. So the correct answer will be, it is the number C, this thiazole ring. So this is the structure of bleomycin. Okay. So this particular structure includes a bis thiazole ring system which interacts with the DNA. So this is the bis thiazole ring, which is the major intercalating portion, intercalating agents in this structure which intercalates with the DNA. So bleomycin, the mechanism of action, it acting through the binding to DNA, which results in single and double strand breaks following the free radical formation and inhibition of DNA synthesis. The DNA fragmentation is due to oxidation of the DNA bleomycin iron 2 complex, and it leads to the chromosomal aberrations. The cell cycle specific drug that causes accumulation of the cells in G2. So once the structure becomes intercalated, the nitrogen atoms of the primary amines and the pyrimidine ring. Okay, so these are there are so many primary NH2 groups here. Also there we have primary amines. We have the NH2 group in the pyrimidine ring. So as you can see, the structure contains various heterocyclic rings. There is a pyrimidine ring, there is a imidazole ring, and no, it also has this bithiazole rings. Okay, so once the structure becomes intercalated, the nitrogen atoms from the primary amines and the pyrimidine ring, the imidazole ring, chelates, ring chelate and ion, that is ferrous ion, okay, the iron Fe2, which then interacts with the oxygen and oxidized to ferric ion. Now during this process, oxygen picks up one electron and forms superoxide, and superoxide can subsequently get converted to the hydrogen peroxide, which then eventually forms the hydroxyl radical. These together are known as the reactive oxygen species, and the cell is very efficient of getting rid of this sum of this reactive oxygen species. However, since the generation of the reactive oxygen species occurs near DNA strand, this can result in the DNA strands being cut particular between the purine and pyrimidine nucleotides. Bleomycin also appears to prevent the enzyme DNA ligase from repairing the damage caused, and whenever there is a damage that occurs in DNA, there are a number of enzymes that come and help the repairing the damage that is caused. Okay. So This was the mechanism of action of the bleomycin, and this is the structure. The structure seems to be very complicated, so you don't need to memorize the structure, but just remember that it has a the dithiazole bithiazole ring, which is important for intercalating, and also it has the different heterocyclic portions. And what is the role of this particular rings and particular groups, which you need to remember. Okay. Now problem number 10, it says that etoposide is number A, intercalating topoisomerase poison, number B, non-intercalating topoisomerase poison, number C, intercalating topoisomerase inhibitor, and number D, non-intercalating topoisomerase inhibitor. This is the structure of etoposide. Okay, this is the structure of etoposide. Now you have to identify the correct answer. Number A, it is what is this etoposide? Number A, intercalating topoisomerase poison. Number B, non-intercalating topoisomerase poison. Number C, intercalating topoisomerase inhibitor. And number D, non-intercalating topoisomerase inhibitor. Okay, once again. Okay, so there is a mistake in the numbering. It will be problem number 8, not problem number 10. So I think there is a mistake. I will just correct it later. So problem number 8 it is, which says etoposide is which kind of agent? Let me know in the chat box, A, B, C or D, which of the following is the correct answer? Let me check the chat box now. Okay, I have answers which says it's number C, D and B. Okay, so I have three different options as number. Okay, B, C and D. Let me check which of the following will be the correct answer. So there is a mistake in the numbering which I will just correct it later. So etoposide is number B, non-intercalating topoisomerase poison. So, the anti-cancer agent etoposide and teniposide, they belong to a class of the compounds known as the podophylotoxins. So, these are semi-synthetic derivatives of the epiphylotoxin and an isomer of naturally occurring agent called the podophylotoxin. So now both of these agents act as topoisomerase poisons. Okay, so DNA strand breakage by this class of molecules is thought to be occur by the free radical process that involves the 4' phenolic group, which is this one. And what happens on what is supposed to happen is the 4' phenolic group forms a semiquinone radical, which is then generate again the reactive oxygen species. Evidence for this comes that if you remove this and if you are able to derivatize this oxygen with a methyl, then the compound becomes inactive. The presence of the glucoside sugar moiety also increases the ability of this molecule to induce the brakes. Okay, so what happens? What is the mechanism of action? That this particular compound, it also induces the reactive oxygen species by the semiquinone structure of the because of this para OH group. And how to prove it? That if you methylate this OH group, then this compound becomes inactive. Okay, and there is a glycoside sugar here also, which increases the ability of this molecule to induce the brakes. Okay, so just let me correct it. I think there are some issues. Okay, so it was the problem number 8. Now I will go to the next problem. There is also mistake in numbering. So it is a problem number 9. So it says that which of the following statements is true regarding the cisplatin? Your options are: number A, this disrupts the base pairing of the adenine and guanine. Number B, in the interstrand cross-linking of DNA results from the action of the drug. Number C, base pairing of cytosine and guanine is disrupted by this. And number D, it has the tetrahedral geometry. Which of the following statements is true regarding cisplatin? Number A, this disrupts the base pairing of adenine and guanine. Number B, interstrand cross-linking of DNA results from the action of this drug. Number C, the base pairing of cytosine and guanine is disrupted by this. And number D, it has a tetrahedral geometry. Which of the following statements is true regarding the cisplatin? Which of the following statements is true regarding cisplatin? Identify the correct statements regarding the cisplatin. Let me check the chat box now. Mhm. I have some answers. All are saying that it is number B. Let me check if number B is the correct answer or not. So the correct answer will be, it is number C. Okay, the statement that is true for the cisplatin is the base pairing of the cytosine and guanine is disrupted by the cisplatin. This statement is true. Okay, so now let us check why this statement is true. So this is the structure of the cisplatin, and also it has a structural analog which is known as the transplatin. Okay, so in cisplatin we have the platinum in the center of the square planar geometry, and in the cisplatin we have this amine NH3 groups and the Cl groups in the cis position. This NH3 NH3 are in the cis position and chlorine chlorine in the cis position, and in the transplatin we have this trans position. So this has a square planar geometry. That is why the statement number 3 is not true, which says it has a tetrahedral geometric. Okay. So basically, what does this particular cisplatin do? So we have the cisplatin here in the blood. So where the chloride ion concentration is 104 millimolar. Okay, but in the cytoplasm, the chloride ion concentration is less, which is the up to 4 to 10 millimolar. Okay, so when the cisplatin enters the cell in the cytoplasm, what happens? These chlorine groups are removed, and we have this water takes in the chlorine position, and we have this species. Okay, and this cisplatin binds with the CG base DNA base pair. Okay, so this is the CG DNA base pair, and we have this nitrogen here in the N7 position of guanine where the cisplatin binds. Okay, and this cisplatin, this causes the 1,2-D GPG intrastrand adduct, which is the major adduct. Okay, so here in the option number in the option number our B, it says that it interstrand cross-linking of DNA results from the action of the drug, but major adduct which is formed, the major cross-linking which is formed, that is intrastrand. Okay, intra, not inter. Intrastrand cross-linking happens due to this action of the cisplatin. Okay, so the first statement is not true, which says that it disrupts the base pairing of adenine and guanine, which is not true. Okay, there is no base pairing in between the adenine and guanine. And then it says the interstrand cross-linking of DNA results from the action of drug, but it will be intrastrand cross-linking of DNA results from the action of the drug. Okay, now number C, which says that base pairing of cytosine and guanine is disrupted by this, which is correct. Okay, and the number D, which says that it has a tetrahedral geometry, it is also not true because it has a square planar geometry. Okay, so this was our problem number 9. Sorry, I made mistake for the numbering of the questions. Okay, so it's fine. So this is the problem number 9. I think it's now clear, right? Why number B is not the correct answer? If you have any doubt, any query, you can let me know in the chat box, okay? So this was our problem number 9. Now I will go to the next problem, which is our problem number 10. Here also the numbering is mistake. Okay, that's fine. So the next problem says, which of the following statements is untrue regarding the mitomycin? Okay, so now you have to find out which of the following statement is not true for mitomycin. Your options are: number A, it is an excellent choice of drug for solid tumors. Number B, it is activated by the bio-oxidative process. Number C, it is a prodrug. Number D, in its mechanism of action, there is a Michael reaction. Which of the following statements is untrue regarding mitomycin? So whenever there is a true, false, true, untrue, or correct, incorrect type of question, read the options very carefully. Most major the student they made mistake in this kind of question because this sometimes the options can be little confusing. So read the options very carefully and then answer this kind of question. Okay, which of the following statements is untrue regarding the mitomycin? Your options are: number A, it is an excellent choice of drug for the solid tumors. Number B, it is activated by bio-oxidative process. Number C, it is a prodrug. And number D, the mechanism of action. In the mechanism of action, there is a Michael reaction. Which will be correct answer? Means which will be the which statement is the not correct? Which of the following statement is untrue regarding mitomycin? Let me check the chat box. Okay, I have few answers. So I have two answers. One is number A and another is number C. Okay, let me check which of the following will be the correct answer. So the statement which is not true regarding mitomycin will be, it is number B. It is activated by bio-oxidative process. This statement is not true. Okay. So the mitomycin was discovered in 1950s and the agent is being isolated from the class of microorganism known as the streptomyces. It is one of the most toxic anti-cancer agents in clinical use, an example of a prodrug. Of course, we will look through the products later, but it's an example, excellent example of a prodrug because and what it does is own way is not because its own is not it is inactive, and when it is converted to the alkylating agent in the body. Okay, so the way it happens is that there are enzymes which carry out what is known as a bioreductive process. Okay, so the option number B, which says that it is activated by the biooxidative process, it is not true because it is activated by the bioreductive process. So there are many enzymes inside our body which can do the oxidation as well as reduction, and these are known as the oxidoreductases. So when mitomycin encounters a bioreductive enzyme, this quinone ring here. So let us first check out what is the structure of mitomycin. So this is the structure of the mitomycin. Okay, so we have this quinone structure here, and also we have this kind of the three-member ring here also. Okay, so when this mitomycin, which is a prodrug, it encounters a bioreductive enzyme. What it happens, like this quinone goes up to the reduction to form a this quinol structure. Okay, that's diol structure, and then now this quinone is the electron withdrawing group. We will imagine this carbonyl group at the electron withdrawing, whereas the diol, this diol is fairly electron rich, which can promote the rearrangement. Okay, so this is the rearrangement which happens here. After this formation of the diol, this rearrangement happens here. Okay, and this rearrangement eventually pick up the proton to produce a species, then becomes a substrate for attack by DNA. As the reduction step is involved in the mechanism, it has been proposed that the drug should be more effective against the tumor in an oxygen-starved or hypoxia environment, such as the center of the solid masses, okay, or the solid tumors. Okay, so this is the mechanism of action. First is the reduction, followed by the rearrangement, which opens up this three-member ring. Then after this three-member ring opening, what happens? This DNA has this amine NH free NH2, which interacts with this alkylating, so it becomes a strong alkylating agent and forms adapt with the DNA. Okay, and then it cross-links with the DNA. And this particular reaction, this particular mechanism also is a on one type of the Michael addition reaction or the Michael kind of rearrangement. Okay, so why this kind of a reagent or this particular agent is active against the solid tumors? Because all this reaction takes place in the reductive environment, right? So if there is oxygen, this mechanism won't be activated. So in the center of the solid tumors, there is an oxygen deprived environment where this particular alkylating agent will be most effective. Okay, so this was the problem number 8. One more thing, just I have to just correct it over here. So it is the, sorry, it is not, it is the problem number 10. Okay. Okay, so now we will go to the next problem. So next problem, it says that which reaction is involved in the method of action of the calcimicin? Your options are number A, Bally-Shillman reaction, number B, Suzuki coupling reaction, number C, Diels-Alder reaction, number D, Boltzmann cyclization reaction. Which of the following reaction is involved in the method of action of the calcimicin? Number A, Bally-Shillman, number B, Suzuki coupling, number D, Diels-Alder, number D, Boltzmann cyclization. Let me know in the chat box A, B, C or D which of the following is the correct answer. This is the problem number 11. So again we have a mistake in numbering, but it's fine. Which reaction is involved in the method of action of calcimicin? Bally-Shillman reaction, Suzuki coupling reaction, Diels-Alder reaction or Boltzmann cyclization reaction? Which reaction is involved in the method of action of calcimicine? Let me check the chat box now. So I have, I think, one answer only, which says number C. Let me check if number C is the correct answer or not. So the reaction which is involved in the method of action of calcimicine is, it is number D, Bergman cyclization reaction. Okay. So what is the Bergman cyclization reaction? So basically we have a indiene, means we have a triple bond followed by a single bond, double bond conjugation, then also single bond, then we also have a triple bond. Okay. So, so this is known as the indiene. Okay. Now, so this indiene, so when there is a heat or H nu, so what happens? There is a cleavage of here and forming a biradical, diradical. Okay. So it is 1,4-benzyl diradical is formed here, and then what happens? This 1,2-benzyl diradical, it abstracts a proton from another ring. Okay, and then what happens? It forms this kind of 6-member ring. So this is the Boltzmann cyclization reaction, which was invented in the 1972. So this is the structure of the calcimicine, okay, which also have this n-diene structure. And first the nucleophile comes here and it attacks the sulfur group here and forms a thiol ring. Then this thiol group lone pair attacks to this position and forms a oxygen minus here. Okay, and then what happens? There is a Boltzmann cyclization, which then cyclize this position. Okay, and we have this phenolic diradical. And what happens is diradical, it interacts with the DNA to form DNA diradical. In presence of oxygen, we have the DNA double strand cleavage. Okay, so this is the mechanism of action of the calcimicine and where the Boltzmann cyclization is involved. So this particular reagent, which is the calcimicins, are also known as a chain cutter. So chain cutters cut the strands of the DNA and prevent the enzyme DNA ligase from repairing the damage. They appear to act by creating diradicals on the DNA structure. These diradicals react with the oxygen to form the peroxy species and the DNA chain fragments. Calcimicin gamma 1 is an example of the chain cutter. So this compound binds with a minor group of DNA and cuts the DNA chain. The reaction starts with a nucleophile attacking the trisulfide group. The thiol, when is free, undergoes a intramolecular Michael addition with a reactive alpha beta unsaturated ketone. The resulting intermediate then cycloaromatize the reaction known as the Boltzmann cyclization to produce an aromatic diradical species which snatches 2 protons from the DNA. As a result, the DNA becomes a diradical and the reaction with oxygen then leads to the chain cutting. Okay. So calcimicin, it is an example of chain cutter. This is the mechanism of action, and this is the there are different reactions are involved. Okay. So this was the problem number 11. Sorry, there is a mistake in numbering again here. I think I will just, yeah. Okay, so now I will go to the next problem. So now, okay, there also again his number is wrong. So it says that acyclovir, the structure is shown here, is an inhibitor of the DNA polymerase selectively in virus infected cell. This is because your options are number A, acyclovir is first converted to monophosphate only by viral thymidine kinase. Number B, acyclovir does not enter the infected cells. Number C, acyclovir first converted to monophosphate only by host thymidine kinase. Number D, acyclovir triphosphate is not accepted by host DNA polymerase. Which of the following is the correct answer of this problem? It says acyclovir is an inhibitor of DNA polymerase selectively in the virus infected cell. So this is because acyclovir, number A, acyclovir is first converted to the monophosphate only by viral thymidine kinase. Number B, acyclovir does not enter the infected cells. Number C, acyclovir first converted to monophosphate only by the host thymidine kinase. And number D, acyclovir triphosphate is not accepted by the host DNA polymerase. Which of the following will be the correct answer? So which of the following will be the correct answer? Let me check the chat box. Okay, I have two answers. One are both are saying number A is the correct answer. Okay, let me check if number A is the correct answer or not. So the correct answer will be, it is number A. Acyclovir is first converted to the monophosphate only by the viral thymidine kinase. Okay. So chain terminators, basically, this kind of acyclovirs are known as a chain terminator. Okay, so normal replication occurs when a triphosphate reacts with the 3' end and forms a phosphodiester bond catalyzed by the DNA polymerase. Acyclovir is an important antiviral drug that was discovered in 1970s and acts as a chain terminator, satisfying all the three requirements. Okay, so this is the structure of the acyclovir. So this is the this is a pro drug. Okay, this acyclovir is first converted to acyclovir triphosphate, which is the particular your active drug, and then the deoxin guanosine it forms. Firstly, it contains a guanine base, which means that it can base pair to cytosine moieties on the template chain. Secondly, although it does not contain a triphosphate group, it is added when the molecule it is added to the molecule in a virally infected cell. Thirdly, the sugar unit is incomplete and lacks the required OH group normally present at 3' position, and the nucleic acid chain does not get extended. Okay, so this acyclovir, it is an example of as a chain terminator. Okay, so now next problem. I will correct the numbering at the last. Okay, so the next problem says that deprivation of adequate oxygen in the tissues are termed as dash. You have to find out the correct term. Your options are number A hypoxia, number B ischemia, number C homeostasis, and number D metabolism. Deprivation of adequate oxygen in the tissues is termed as what? Hypoxia, ischemia, homeostasis, or metabolism? Which of the following will be the correct term? The deprivation of adequate oxygen in the tissues is termed as what? Hypoxia, ischemia, homeostasis, or metabolism? Let me know in the chat box A, B, C, or D which of the following is the correct term for this phenomena. Let me check the chat box. Okay, I have a couple of answers say which is saying the number A is the correct answer. Let me check if number A is the correct or not. So the correct answer is yes, it is number A. Hypoxia is the term that indicates the deprivation of adequate oxygen. Okay, so hypoxia is a condition in which the body or a region of the body is deprived of accurate oxygen supply at the tissue level. Hypoxia may be classified as either generalized or affecting the whole body or local affecting the region of the body. So this is the hypoxia. So basically, what are the regulators? The epigenetic regulation, the genetic regulation, post-transcriptional regulations are also there. Then downstream mediators like this kind, all these markers and all these enzymes, cellular effects, proliferation, survival, secretion of the angiogenic factors, microcebe, vesicle, metabolic education, and pathological outcome is tuberogenesis, angiogenesis, drug resistance, and metastasis. Okay, so hypoxia, this particular deprivation of oxygen is frequently seen in the center of the solid tumors, which I have already mentioned that in this case of so in the center of the solid tumors there are oxygen deprived position, which is the hypoxia position. So other terms which I have already mentioned are the ischemia. Okay, so it is a stage when the tissue or the organ has its arterial perfusion lowered relative to the metabolic needs, or ischemia defined as a condition in the inadequate blood supply in an area of the tissue. Okay, so it is an inadequate blood supply in the part of the body even to the point of the complete deprivation, and is simply okay. Then we have the homeostasis. This term was also mentioned. So homeostasis is the condition in which the internal environment of the body remains relatively constant despite changes of the external environment. Examples would be the maintenance of body temperature and the levels of the glucose in the blood. And we have the metabolism, the definition, sum of all chemical reactions in the body, anabolic versus catabolic reactions. Anabolic reactions use the chemical energy to synthesize products, and catabolic reaction breakdown of the substances to generate the chemical energy. Okay. So now we will go to the next problem. So it says that which of the following is known as the antigenic determinant? Your options are number A hapten, number B epitope, number C enzymes, and number D paratope. Which of the following is known as the antigenic determinant? Antigenic determinant. Which of the following is known as antigenic determinant? Hapten, epitope, enzymes, or paratope? Which of the following is known as an antigenic determinant? Hapten, epitope, enzymes, or paratope? Let me check the chat box. Hmm. So I have two options. One is saying number A, another is saying number B. Let me check which of the following will be the correct answer. So the correct answer will be It is number B. Epitope is known as the antigenic determinant. So what are the, now let us see what does this H and every term means. So hapten is a molecule too small to stimulate the antibody formation by itself. However, when the hapten is combined with a larger carrier molecule, usually a serum protein, the hapten and its carrier together form a conjugate that can stimulate an immune response. Okay, so these are the hapten molecules, this is a carrier molecule, and hapten carrier conjugate, they can stimulate an immune response. Then we have abzyme. So abzyme is an form antibody, and enzyme is often called as catalytic antibody, which is a monoclonal antibody with the catalytic activity. Okay, so this is the structure of the abzyme. We have okay. Then we have this epitope. Epitope is also known as the antigenic determinant, is a part of an antigen that is recognized by the immune system, specifically the antibodies B cell and T cells. And also we have paratope. Paratope is a part of the antibody which is recognizes the antigen binding site of the antibody. Okay, this is a small region of 15 to 22 amino acids of the antibodies. Every region that contains a part of the antibodies heavy and light chain. So this is the structure of the antibody. The antibody has the paratopes, okay, which recognizes the epitopes of the antigen. So antigen has this antigenic determinant, which is a part of antigen, okay, which is recognized by the antibody. Okay, so remember this is the paratopes is a part of the antibody which recognizes the epitope, which is a part of the antigen, and together they form this antigen antibody conjugate. Okay, so correct answer was number B. Now next problem, it says which of the following statements is true regarding antibodies? Your options are number A, T cells are associated with the humoral anti-immunity. Number B, B cells are associated with the cellular immunity. Number C, B lymphocytes are present in the bone marrow. Number D, B lymphocytes generated by the thymus. Which of the following statements is true regarding antibodies? Number A, T cells are associated with the humoral immunity. Number B, B cells are associated with the cellular immunity. Number C, B lymphocytes are present in the bone marrow. And number D, B lymphocytes are generated by the thymus. Which of the following statements is true regarding the antibodies? Let me know in the chat box A, B, C or D which of the following statements is true regarding antibodies. Your option number A says T cells are associated with the humoral immunity. Number B, B cells are associated with the cellular immunity. Number C says B lymphocytes are present in bone marrow. And number D says B lymphocytes are generated by thymus. Let me check the chat box. Okay, I have two answers A and C. Let me check which of the following will be the correct answer. So the correct answer is number C. B lymphocytes are present in bone marrow. This statement is true. So what does this B lymphocytes and T lymphocytes do? So basically, B cells or B lymphocytes, they attack the inhibitors, the invaders outside the cells. Okay, so and T cells, they attack the infected cells itself. Okay, so B lymphocytes origination and maturation takes place in bone marrow, Okay, and T lymphocytes origination takes place in bone marrow, but the maturation in the thymus. So this B lymphocyte they differentiate into the plasma cell. T lymphocyte differentiates into plasma cell or memory cell or cytotoxic T cells. B lymphocytes are involved in the humoral immunity, and T lymphocytes are involved in the cellular immunity. The B lymphocytes they produce the antibody, and the B T lymphocyte they help the B lymphocyte to produce the antibodies. Okay, so these are the differences and comparisons between the B lymphocytes and T lymphocytes. Okay, so remember that B lymphocytes they basically destroy the foreign species outside the cell, but the T cells basically they kills the infected cells. Okay. Now I will go to the next problem. So it says that which of the following statements is false for the primary metabolites? Your options are number A, these are a part of the basic molecular structure of an organism. Number B, vitamins are an example of the primary metabolites. Number C, they are not directly involved in the growth and development and reproduction. And number D, these are produced in the large quantities. Which of the following statements is false for the primary metabolites? Your options are number A, these are the part of the basic molecular structure of an organism. Number B, vitamins are an example of primary metabolites. Number C, they are not directly involved in the growth and development and reproduction. Number D, these are produced in the large quantities. Which of the following statements is false for primary metabolites? Which of the following statements is false for primary metabolite? Let me check the chat box. Okay. So I have three options. One is not saying it is number C, and another is saying number two are saying number D. So C or D. Let me check which of the following statement will be correct. Okay. So the statement which is false regarding primary metabolites are, it is number C, which says that they are not directly involved in the growth, development, and reproduction. These statements is false. Okay. So what are the primary metabolites? So these are the chemical compounds produced during the growth and development processes. They are also involved in the primary metabolic processes of respiration and photosynthesis. The primary metabolites are formed in the growth phase. They maintain the physiological functions of the body and are known as the central metabolite. They are intermediate products of the anabolic metabolism, which are used by the cells for the formation of essential micromolecules. Amino acids, vitamin, organic acids are some of the primary metabolites produced industrially. Alcohol is the major primary metabolite produced in the large scale industrially. So what are the differences between the primary and secondary metabolites? Primary metabolites perform the physiological functions in the body like intrinsic functions. Secondary metabolites are the derivatives of the primary metabolites. Primary metabolites are directly involved in the growth, development and reproduction and formed during the growth phase due to the energy metabolism. Secondary metabolites, they play a role in the ecological functions and formed near the stationary phase of the growth. The primary metabolites produced in the large quantities and the extraction is easy increasing, and the secondary metabolites produced in the small quantities and extraction is difficult. The primary metabolites are same for all the plants, but unique for the secondary ones are the unique for the different plant species. Primary metabolites are a part of a basic molecular structure of the organism. The secondary metabolite not a part of the basic molecular structure of the organism. Ethanol, lactic acid, nucleotide, vitamins are the examples of the primary metabolites. Pigments, antibiotics, and drugs are the examples of the secondary metabolites. Okay. So number C, the statement was the false regarding the primary metabolites. Now I will go to the next problem. So it says that pyridoxal phosphate is a coenzyme of dash. Your options are number A, aminotransferase, number B, glutaminase, number C, glutamine synthase, and number D, glutamate dehydrogenase. Pyridoxal phosphate is a coenzyme of what? Number A, aminotransferase, number B, glutaminase, number C, glutamine synthase, number D, glutamate dehydrogenase. Let me know in the chat box A, B, C or D which of the following is the correct answer. Pyridoxal phosphate is a coenzyme of what? Let me check the chat box. Okay, so I have few answers. Number A is the only answer is given. Let me check if it is the correct one or not. So pyridoxal phosphate is coenzyme of, yes, number A, aminotransferases. This is the structure of the pyridoxal phosphate or PLP. So what is the mechanism of action? The metabolically active form of the vitamin B6 is the pyridoxal phosphate, which serves as a coenzyme of numerous enzymes, most of which are involved in the metabolism of the amino acids. Okay, the general mechanisms are the decarboxylation, transamination, racemization, elimination, or replacement reactions, beta group interconversions also. So this is the pyridoxal phosphate. It has another pyridoxal phosphate which has another active form, another form which is the pyridoxine phosphate. So there is a amino donor, which is substrate amino acid. So what does it do? Basically, it transfers the amino group. That is why the name is the aminotransferases. Okay, so this is a substrate amino group which has the amino group. Then first, what does this pyridoxal phosphate do? It takes up the NH3 group. Okay, from this donor amino group, and then it transfers it to a amino acceptor, which is a substrate oxoacid, to form a product amino acid. Okay, so this is the reaction which is catalyzed by this pyridoxal phosphate. And remember that aminotransferases is are the major. This pyridoxal phosphate is a coenzyme for the aminotransferase enzymes. Okay, which basically transfers this amine groups. Now I will go to the next problem. So this is the problem number 18, which says that correct sets of three coenzymes involved in the biosynthesis of acetyl-CoA are number A, TPP, FAD and NADH, number B, TPP, lipoic acid and FAD, number C, FAD, NAD plus and lipoic acid, and number D, lipoic acid, NADH and TPP. So what is the correct set of three coenzymes involved in the biosynthesis of acetyl-CoA? Your options are number A, TPP, FAD, NADH, number B, TPP, lipoic acid and FAD, number C, FAD, NAD plus and lipoic acid, and number D, lipoic acid, NADH and TPP. Correct set of three coenzymes involved in the biosynthesis of acetyl-CoA. What will be the correct answer? Let me know in the chat box, A, B, C or D. What will be the correct set of the coenzymes involved in the biosynthesis of acetyl-CoA? So what will be the correct sets of three coenzymes involved in the biosynthesis of acetyl-CoA? Let me check the chat box. Okay, number A. I think this number A is the for. Okay, I have a different answer. Number B, number A, and number A. Okay, let me check which of the following is the correct answer, B or A. So the correct answer will be, it is number B. Okay, TPP, lipoic acid and FAD, this is the correct set of the coenzyme that is involved in the biosynthesis of acetyl-CoA. So basically, conversion of this pyruvate and acetyl-CoA, it takes up the help of these all this coenzymes. Okay, so what does we have? So this is a net reaction is oxidative decarboxylation of pyruvate. First carbons of the glucose is fully oxidized. So this catalyzed by PDH is required 5 coenzymes. Okay, TPP, this lipoate and FAD. These are the prosthetic groups. Okay, and then NAD plus and CoSH, SH, they are the co-substrates. Okay, so these are derived from the TPP. This the derived from the so TPP is derived from the thymine, FAD is from riboflavin B2, NAD is niacin B3, and CoA from the plantothenic acid B5. So this is the structure of the pyruvate, and this is the structure of the acetyl-CoA. So as you can see, this is basically the decarboxylation reaction, which is oxidative in nature, and pyruvate dehydrogenase complex is involved here. Okay, and we have the coenzymes TPP, lipoate and lipoic acid or lipoate and FAD here, and total delta G is that minus 33.4 kJ per mole. Okay, so number B was the correct answer. Problem number 19, it says what are the main enzyme systems for the catalyzing of the ethanol to acetaldehyde? Your options are number A, ADH, number B, ADH, CYP2E1 and catalase, number C, CYP2EA, and number D, catalase. What is or are is the main enzyme for the catalyzing ethanol to acetaldehyde? Number A, ADH. Number B, ADH, CYP2E1 and catalase. Number C, CYP2E1 and number D, catalase. Which of the following is the correct answer for problem number 19? A, B, C or D? Let me know in the chat box. What will be the main enzyme system for catalyzing ethanol to acetaldehyde? Let me check the chat box. Okay, so I have two answers. Both are saying it's number A. Okay, let me check if number A is the correct answer or not. So the correct answer is it's number B. ADH, CYP2E1, catalase, all three are involved in catalyzing the ethanol to acetaldehyde. Okay, so basically this ADH, it is the alcohol dehydrogenase enzyme, okay, which is involved in the oxidation reaction or redox reaction. So only one of the protons of this ethanol is abstracted and added to the specific site of the NAD plus, okay, and then we form this NADH is formed by the enzyme alcohol dehydrogenase. So alcohol dehydrogenase or ADG it is an NAD plus dependent cytoplasmic enzyme. It oxidizes ethanol to acetaldehyde. ADH is a dimer and has a six isoenzymes. It has some individual enzymes are mutated. In the individuals, alcohol metabolism is slower and even small quantities of alcohol may be produced symptoms of intoxication. So ethanol to acetaldehyde. So what are the enzymes that are involved in the whole process? So ADH is involved. Okay, it basically ADH can involve and also catalase it can also it can also convert the ethanol to acetaldehyde, okay, by converting the side reaction is H2O2 to H2O, and then also CYP2E1 the side reaction is NADPH to NAD plus, okay. So basically this all these three enzymes can catalyze this ethanol to acetaldehyde conversion. In case of this ADH, the side reaction is the conversion of NAD plus to NADH. In case of CYP2E1, the side reaction is the oxygen plus NADPH to NAD plus to H2O. And in case of catalyst, is the H2O2 to H2O. Okay. So I think the question is little bit, it should be like this. Which of the following? Yeah, I think I should have rephrased this particular question because the question seems to be a little difficult to understand. Okay. So basically, it is asked that which of these following enzymes is responsible for this particular conversion. ADH is responsible, but catalase and CYP2E1 are also can do this conversion. Okay. So now I think the question is more correct or more understandable. So now the last question of today's session, it says that the following structure is used in the treatment of the brain tumors. What is the structure called? Your options are number A, carmustine, number B, lomustine, number C, streptozotocin, and number C, cyclophosphamide. What is this structure called? This particular compound or therapeutic is used in the treatment of the brain tumors. What is this the structure calls? Your options are number A, carmustine, number B, lomustine, number C, streptozotocin, and number D, cyclophosphamide. What is the structures called? So what is this following structure called? Let me check the chat box. Yeah, the previous question was not correctly framed. Actually, I was just wanted to ask that which of the following enzyme was involved in the conversion of ethanol to acetaldehyde. Okay. So all the three, so that question was a little confusing. I agree. So that is why I have changed the question now. Okay, we can check the updated PPT when I upload this PPT. And for the last question, I think I have only one answer, which says it is number A. Let me check. Okay, so I have rephrased this problem number 19, which says that what is or are the main enzyme for catalyzing ethanol to acetaldehyde. Okay, so all these three, so now I think this is more correct answer because all these three enzymes are involved in the catalyzing ethanol to acetaldehyde. Okay, alcohol dehydrogenase I have mentioned in detail, but others are also involved in this particular conversion. So all three will be the correct answer. So problem number 20, the correct answer will be, it is number B, lomustine. This particular structure is known as lomustine, which is a treatment used for the treatment of the brain cancer. Okay, so it is an alkylating nitrosourea compound used in chemotherapy. It is closely related to the semustine and is in the same family of the streptozotocin, but it is a highly lipid soluble drug. Thus, it crosses the blood-brain barrier. Okay, the property makes this ideal for treating brain tumors, which is its primary use. Although it is used to treat the Hodgkin lymphoma as the second line option. So this is the structure of the carmustine, which works by slowing or stopping the cancer cells. This is the structure of the streptozotocin antibiotic that produces pancreatic islet beta cell destruction. This is the structure of the cyclophosphamide used to treat cancer of the ovaries, breast, and blood and lymph system. Okay. So all the structures I mentioned along with their mechanism of action. So this is the structure of the lomustine, which is the therapeutic for the brain tumor, treatment of the brain tumor. So this was the last question of today's session. So today what we did, we discussed about the different the compound or different therapeutic agents which targets the nucleic acids. Okay, we discussed about the topoisomerase enzymes, inhibitor, topoisomerase poison, intercalating agents, chain cutters. Okay, also we discussed about the chain terminators and also on, and then we discussed a little about the alkylating agents like your cyclophosphamide. They followed by the discussion regarding to the various biological systems. Okay, the antigen antibody, this particular things and metabolites. So we already discussed today's class. So this presentation and the this class recording I will be uploading in the NPTEL website soon. So where you have to find it, then you can go to the medicinal chemistry course outline. So this is the course outline of medicinal chemistry. Okay, there you will find in the left panel there is a problem solving session of January 2025. There, if you just click on here, it will be directly, if you click here, you will be directly redirected to a page where you can find the class recording and also also the PPT. So now I will share my email ID here in the chat box. Okay, please let me know if you have any doubts or any query regarding today's class or regarding any previous classes. So this is my email ID. If you have any doubt, any query regarding today's course content or regarding any course content which we have previously discussed in the past 4 weeks, you can let me know through the chat box. Okay. Now, okay. So if you can let me know through this email. You can just save this email, and this is our week 5 live session. And the next week we will meet with the week 6 course content. We will discuss the week 6 course content in our next class, which will be on next Friday from 6 p.m. to 8 p.m. So if there is no problem or no queries, I will close the session for today. So is there any doubts or any query for today's class? If there is any problem, you can feel free to reach me through the email ID, okay, which is given here. Otherwise, I will close the session for today. So thank you all for joining. I will be uploading this class material, and also I will uploading this class recording in the NPTEL website. Please check it out from there. And if you have any query, any doubt, feel free to ask me. Okay. Thank you all for joining, and thank you all for answering the questions very actively. I will meet you in the next class, which will be on next Friday. So thank you all. I will close the session for today.